My presentation today will be looking at the impacts of fetal growth restriction on the brain and using basically the large animal model of fetal growth restriction, which is the piglet. So just a really brief background, fetal growth restrictions where a baby fails to grow to its full potential whilst developing in the womb, and it's commonly caused by placental insufficiency where there's a reduced nutrient and oxygen transport to the developing baby in utero. It's the second leading cause of perinatal death and long-term disability after prematurity, and the developing brain is quite vulnerable to this condition, with adverse outcomes including learning difficulties, attention difficulties, autism, ADHD, and cerebral palsy. So up to half of these infants are reported to have neurodevelopmental delays at 2 years of age. So you can see it has quite an impact on these children's lives and also their families and the society. But unfortunately, there's no treatment available to protect the growth-restricted newborn from these long-term adverse outcomes. So this is where we move to animal models to be able to explore mechanisms of brain injury and then hopefully provide treatment options for these vulnerable newborns. So why do we use the piglet to examine adverse perinatal brain outcomes? There are quite a number of reasons. So firstly, the human and the piglet have similar gyral complexity. So having a look down the bottom here, you can see the quite smooth surface of the rat brain in comparison to the highly folded piglet brain and the human brain. They also have similar grey to white matter ratio. And additionally, brain growth spurt in the piglet is similar to the human. So looking at this graph here is birth over the time course. And if you track the human, you see they peak at birth. And then if you track the piglet, they peak at birth too. Unlike the monkey or the sheep or the guinea pig, where their brain growth spurt is prenatally, or the rat or the rabbit, whose brain growth spurt is postnatally. So we feel that's really an ideal model to examine altered brain development arising from compromising perinatal events. So now looking at the growth-restricted piglet, around 15 to 20% of piglets in each litter are growth-restricted. And this actually occurs spontaneously. So we don't need to use any physical or chemical or nutritional interventions like other animal models of fetal growth restriction would do, which minimises the confounding impacts of experimental interventions. It also mimics the human situation really well, with asymmetric growth restriction resulting in brain sparing. And this occurs in around 70 to 80% of the human growth restricted population, where this fetal circuitry redistribution occurs where there's more shunting to the brain and the heart compared with all the peripheral organs. So we're able to identify these growth restricted piglets by their lower birth weight and also their head shape. So with the growth restricted piglet, they had this sort of dolphin-like head shape with bulging eyes and wrinkles perpendicular to the mouth. And these are a couple of our little piglets here. So we have the normally grown on the left and the growth restricted on the right. And you can see it has quite a similar bulbous head to this image here. We can also identify them by the less than 10th centile for their birth weight, brain to body weight ratio, and brain to liver weight ratio at post-mortem. So human imaging studies have shown a reduction in grey and white matter volume in FGR neonates, and these alterations have been associated with developmental disabilities. So we know that there's neuronal and white matter injury in the brain, but what we need to do is use animal models to explore which specific cells are vulnerable to growth restriction, when it actually commences, and the mechanisms of injury, so then we can provide these therapeutic interventions. So we've seen neuronal disruption in our growth restricted piglet. So if I point you down to the bottom of the screen here, we've used quite a few different time points to examine the brains in growth restricted piglets. So this first time point was 100 days gestation, and so full term is 115 days. So it's equivalent to around 26 weeks in the human. So when we had a look at microtubule associated protein, which labels for cell bodies and dendrites, we didn't actually see any differences at this time point at 100 days gestation between the growth restricted and the normally grown piglet. However, when we examined the brains at 104 days gestation, which is approximate to around 28 weeks, so coming into the third trimester, This is where we're starting to see a difference between the normally grown and the growth restricted in the neuronal cell count. And it continued all the way to our last time point that examined at postnatal day 7. So having a look over here at this image, we labelled mature neuronal cells using UN, which is in the green, and MAP2, which is the structural neuronal marker labelling the cell body and the dendrites, in the red. We have a normally grown brain on the left here and the fetally growth restricted here. So you can see that there's quite a few regions with sparse labelling throughout the cortex in the growth restricted brain. And when we count these, so we have normally grown in the blue and fetal growth restricted in the red. So there's a significant reduction in MAP2 labelled area, as well as significant reduction in UN cell counts. But not only are we seeing this reduction in the cell counts, we're actually seeing a difference in the morphology. So if you look over here, we have UN again in green. So we have a control brain here and a growth restricted brain here. And you can see that they're labelling quite different. The cell bodies don't look as plump as they do in the control brain. So in addition to this reduction, there's definitely a morphological difference to the neurons in the brain. We're seeing a reduction in cellular proliferation using Ki-67 here, and also an increase in cellular apoptosis using caspase-3 indicated here. Oops. Sorry. We're also seeing white matter disruption in the growth restricted piglet. So again, using exactly the same time points as we use for the neuronal markers, we're seeing, when we look at myelin basic protein, we're seeing no difference between the normally grown and the growth restricted at 100 days gestation. However, from 104 days gestation up until at least postnatal day 7, we see a significant reduction in the myelination when we count it using myelin basic protein. Another marker we used to look for white matter was Luxol fast blue. So we have a normally grown brain up the top here and a growth restricted down the bottom. And both at postnatal day 1 and postnatal day 4, we see a significant reduction in the myelin count. And you can see even visually, there's less labelling and it's not as well structured through these tracks through the cortex. So we wanted to have also a look at other cells in the white matter. So we looked at oligodendrocytes as well. So oligodendrocytes produce layers of myelin that wrap around the neuronal axons and act as layers of insulation for fast transmission of information. So when we look at this top panel here through the normally grown, we have MBP labelled in green, neurofilament labelled in red for the axons, and OLIG2 in pink, which is a pan-oligodendrocyte marker. So in the normally grown brain, we can see that when we put MBP and neurofilament, co-localize them, they have really nice, strong co-localization. There's really well organised and consistent labelling across the white matter tracks here. We also see consistent presence of OLIG2 oligodendrocytes throughout these tracks. However, looking at these bottom panels here, this is in the growth restricted brain, we can see disrupted labelling throughout the tracks, and there's a loss of co-localization of MBP and neurofilament, as well as a decrease in OLIG2. And this is also again indicated by these graphs with a significant reduction in MBP labelled area and OLIG2 cell counts. So we're seeing this partial loss of myelination across the axons. There is some common theme that they might be just delayed development, but we're actually seeing apoptosis in the oligodendrocytes, so an increased apoptotic oligodendrocytes. When we labelled for caspase-3 to have a look at apoptosis, we have the normally grown brain up the top here, so OLIG2 is in the red, caspase-3 is in the green, and you can see visually there's a lot less caspase-3 in the normally grown in comparison to the growth restricted. So you can see there's quite a large number of apoptotic cells indicated by green in the growth restricted brain. And this is shown here also with these cell counts of caspase-3 positive OLIG2 cells. We're also seeing a loss of OLIG2 is associated with lower myelination. So here we have OLIG2 cells across the bottom and myelination MBP stained area along here. And you can see there's a significant correlation there. When we visually have a look at the white matter tracks and quantitate the dispersion, we can see here with the normally grown and growth restricted, there's more dispersion and varied orientation of these tracks through the growth restricted brain. So this decreased and disorganized myelination of white matter tracks may contribute to the long-term white matter alterations that have been seen in MRI studies in the humans. Again, we're not just seeing oligodendrocyte apoptosis, we're also seeing neuronal apoptosis. So we're seeing high numbers of caspase-3 in the brain. So here we have the FGR brain with caspase-3, but when we co-localized with mature neurons in the grey here, we actually saw limited co-localization. When we had a look at caspase-9 on the other hand, we saw an increase in caspase-9 in the brain, and a significantly increased proportion of neurons are actually undergoing this initiation of apoptosis. So this is indicated over here where in the red we have normally grown counts in caspase-9, and in the growth restricted we see around 66% are undergoing this initiated caspase activation. So the loss in neurons may be attributed to this pro-apoptotic event as a result of potentially pro-inflammatory environment in the brain. We also have evidence of blood-brain barrier disruption in the growth-restricted piglet. So as the name suggests, the blood-brain barrier separates the brain from the blood. So an intact blood-brain barrier is really critical for a healthy brain environment. So the blood-brain barrier makes up many types of cells, but the main ones are that we're looking at are the endothelial cells lined along here, and they're interconnected by tight junctions. So any disruption to this blood-brain barrier can allow the infiltration of toxic substances such as pro-inflammatory cytokines into the brain parenchyma. We can visualize this using a serum protein, so IgG, which shouldn't be evident in a healthy brain. So when we have a look down here in the green, this is a postnatal day 1 growth restricted. You can see that this halo effect is coming from the IgG leakage from the blood vessel out into the brain parenchyma. And it's still evident at postnatal day 4, but minimal or if any leakage in the control brain. When we hone in and look at a little closer at the blood vessels, we use CD34 to have examine endothelial cells. So these are in the red in the top panel here. And you can see that there's less CD34 labeling in the growth restricted brain, which indicates fewer blood vessels and suggesting blood-brain barrier damage. So astrocytes are really important at the at blood vessels because they wrap their end feet. They're all in contact, maybe 99% of them are in contact with the endothelial cells, and they act as a maintenance and stability at the blood-brain barrier. So here in this brain here, we can see CD34 again for the blood vessels labelled in red, GFAP in green for the astrocytes. So you can see the contact here is indicative of the yellow, and they have a nice contact with the endothelial cells in the control brain. Yet when we have a look at the growth restricted brain, we see less astrocytic end feet contact, and the astrocytes actually appear to be in a more reactive state. So having a look at both the astrocytes and IgG leakage in the brain, we had a look and found that when, this top panel here is growth restricted. Well, actually, they're all growth restricted, but when we got in and had a really good look at where the astrocytes were, when they weren't in contact with the blood vessels, we could see all this leakage coming out into the brain of the IgG. But when the astrocytes were in contact, there was minimal leakage into the brain parenchyma. We also used another serum protein called albumin, and this is in the blue. And so the astrocytes in the green here, you can see when they're in contact, they're keeping it in the luminal or perivascular space, but when there is minimal contact of the astrocytes, the albumin is leaking into the brain parenchyma here. We're also seeing another pattern of albumin. It's been taken up into neurons, right near the blood vessels in the brain parenchyma, but we don't know if they're in a apoptotic state. So we're still investigating that at the moment, what that may mean. We're further seeing immune cell infiltration into the growth-restricted piglet brain. So using the pan T cell marker CD3, labelled in green here, we see quite overt infiltration into the brain parenchyma, into the lumen, the perivascular space in regions in the growth-restricted brain. So this is the growth-restricted brain here. Astrocytes are labelled in pink. This is a normally grown here. You can see there's a lot of astrocytes all around the blood vessels, and the T cells are remaining in the vessel lumen. But when there's minimal astrocytic contact, you're seeing these T cells coming out into the brain parenchyma. So, but the thing is, we're just unsure what type of cells these are, whether they're rescue cells, they could be CD4 positive or toxic. Because we're using piglet brain tissue, we do have issues with antibodies. So we're currently looking into this at the moment to see what type of cells these are. Another way to look at neurovascular disruption is, which you would not undertake in a human neonatal cohort, is gadolinium contrast MRI. So when we had a look visually at it, we couldn't see any overt leakage. However, when we undertook a time course analysis, we demonstrated altered handling of gadolinium in the growth-restricted brain. So we looked at T2, which is a vascular, and these are in these top two panels here, in the cortex and in the thalamus. We have normally grown in the blue and growth-restricted in the red. So we see this rapid rise followed by gradual decay. In the T1 tissue, which is the tissue, we saw a rapid rise and maintained plateau, but you can see it's evidently higher in the growth-restricted piglet in comparison to the normally grown. And again, when we quantitate these, you can see the significant difference. So we see a higher level of gadolinium in the vasculature and the tissue of FGR piglets, which correlates with our immunohistochemical analysis of blood vessel impairments. So we've seen that brain injury in the growth-restricted newborn piglet continues after birth, so after the baby's been removed from the adverse environment, which provides us this window of opportunity to treat growth-restricted newborns at birth to improve outcomes. This is really important because even if there were an in utero treatment, around half of these babies are only diagnosed around the time of birth, so it may not even be possible to administer any treatment to the baby in utero. There are very few clinical trials being undertaken in the newborn, so we need to turn to animal studies to have a look at treatments to assess the safety and efficacy of any of these treatments. We recently, in 2018, undertook a literature review, and there are a few rat models looking at postnatal and a couple of antenatal treatments. But more recently, some large animal models of growth restriction are exploring postnatal treatments and finding some really promising results. So the first treatment we were really interested in was ibuprofen. So ibuprofen is a non-steroidal anti-inflammatory drug. It's commonly administered to the preterm neonate to treat patent ductal arteriosus or heart defect. So we found this really small cohort study where they had a preterm infants treated for PDA with ibuprofen actually had preserved MRI volumes compared to those without ibuprofen administration. So this is why we wanted to have a look at it in the piglet. So we used a very similar dosage to what would be used in the human neonate. So that's a 3-day treatment. And we found some nice results looking at recovery to neurons. So this is ibuprofen treatment here, normally grown and growth restricted. Also the white matter. I won't go into too much detail here for time constraints. We saw a reduction in inflammation. So this light bar here is the growth restricted, and the dark red is ibuprofen treatment. So you can see there's quite a dramatic decrease in these pro-inflammatory cytokines in the brain. And we also saw a significant reduction of immune cell infiltrates into the brain. So stem cells have been really getting a lot of interest at the moment. So we had colleagues down in Melbourne are looking at umbilical cord blood-derived stem cells in their growth-restricted newborn piglets, and they're showing some really nice results with a reduction in inflammation in the brain and also recovery to vasculature. We're currently looking at a combination stem cell treatment, which I will discuss in the next slide. So these stem cells that we're currently examining are placental stem cells from the healthy human term placenta. So there's two types, mesenchymal stromal cells and endothelial colony forming cells. So the MSCs have many, many different types of properties, as you can see over here, but one of their major properties is their anti-inflammatory action. ECFCs are true vascular stem cells, so they go up to the blood vessels and migrate there and act to repair on the blood vessels. So our collaborators found that this combination treatment is really effective in immunocompetent animals, so without any immunosuppression, so they weren't rejected. So we're potentially able to use these in an allogeneic situation to protect the newborn brain. We found they worked really well. We administered on day of birth and then assessed 3 days later and saw an improvement to the neurons, to the white matter, reduction in activated microglia and astrocytes. They even worked better than the MSC alone. They were superior to the MSC alone treatment. But interestingly, when we had a look at the pro-inflammatory or the inflammatory cytokines, they didn't have quite an overt effect on the pro-inflammatory, but on the anti-inflammatory they did. So these red is the FGR non-treated and light red or pink is the combination treatment. So you can see there's quite an increase in these anti-inflammatory cytokines in the brain. So we're seeing a really nice response. So what we're currently doing is looking at longer term outcomes. So we just want to see if the safety and efficacy of this treatment. So in conclusion, these piglet studies are providing the opportunity to determine and track mechanisms of brain injury in a clinically relevant animal model of growth restriction. And findings from these studies may be able to provide this critical information we need to rapidly translate neuroprotective interventions to clinic to improve outcomes for babies. And I'd just like to thank all of the team at the Perinatal Research Centre and our collaborators. These are our stem cell collaborators. Professor Paul Colditz, he's a neonatologist and the director of the Perinatal Research Centre, and also our funding bodies who've made this work possible. Thank you.
Thank you, Julie, for very nice presentation on the brain structure in the FGR animal models that your lab use specifically the IUGR piglets, is it? Because it's not that common in Indonesia. We usually use the rodents like that. But, I will try to recap in bahasa a bit for the audience, if it's okay with you. Terima kasih, Dr. Wixey tadi sudah menjelaskan dengan gamblang tentang adanya perubahan struktur baik pada neuron maupun neuroglia pada model hewan coba yang mengalami IUGR atau fetal growth restriction, ya. Jadi berat badan lahir rendah, di mana animal model yang digunakan pada labnya Dr. Wixey ini adalah piglets. Piglets itu anak babi, ya, sus scrova, itu bahasa latinnya itu. Karena apa? Tadi sudah dipresentasikan dengan baik bahwa memiliki beberapa similarity dalam growth spurt, terutama pada brain growth spurtnya itu paling similar dengan trajektori pada otak manusia. Kemudian beberapa teknik tadi yang sudah disampaikan Dr. Julie termasuk menyampaikan dengan menggunakan imunohistokimia ya, fluoresen, beberapa antibody seperti MBP, MAP2 ya, kemudian ada CD34, kemudian ada OLIG2 dan seterusnya. semuanya itu untuk menangkap adanya abnormalitas yang terjadi pada struktur korteks hewan coba suskrova ya piglet yang mengalami fetal growth restriction. Kemudian beliau tadi juga menyampaikan ada beberapa percobaan yang sudah dipublikasikan oleh kelompok beliau di Australia menggunakan antiinflamasi. Karena underlying-nya yang disorot adalah terjadinya inflamasi pada FGR, brain. Jadi beliau tadi pertama menggunakan ibuprofen, hasilnya baik, ya, signifikan tentu saja pada hewan coba ya. Kemudian yang paling terkini adalah menggunakan stem cell. Ada 2 jenis ya, MSC dicampur dengan ECFC. Dan hasilnya cukup signifikan dibandingkan dengan kontrol. Di mana beliau tadi memaparkan kombinasi dua jenis stem cell ini bisa membantu meningkatkan kadar anti-inflammatory agents ya pada daerah otak yang diteliti.